introduction, and thank you for the opportunity to present our latest research results. Today I will present you 3 different research topics we worked on in the last years, and all of them for investigation of microbiomes and host pathogen interactions, where we used nanopore sequencing technology. This works. First I will speak about the microbiome in health and disease. And just to give you a short overview about our microbiome and the microbiota in our body, the human microbiome is made up of around 100 trillion symbiotic microbes. And just for comparison, this is more than we have stars in our galaxy. And the main part, so 95% of this microbiome, is located in the gastrointestinal tract. Because of this, we focus mainly on this area and analyze stool samples of patients to investigate their microbial composition. The human microbiome is not stable. It changes across human life stages. And it begins even before birth with the first immune imprinting. And then we have the primary succession, where we build up our own microbiota, which is like a fingerprint for every person. And then we have this disturbance when we leave our parents' household, because then we have other community-acquired microbiota. And we build a new microbiome, a new specific microbiome for ourselves until the last or the late stages of our life, where again the microbiome changes. So it's very it's not stable. It can change a lot, and it's directly connected to our immune system and the adaptive and innate immunity for our body. We are using long-read sequencing technology for the analysis, so especially Oxford Nanopore Technologie, which you can see here. We are using the MinION and PromethION devices. On the left side, you see this MinION, which is roughly the size of an old large USB sticks. Today they are much smaller, but so we don't need large machines to do our research. And this technique works. So you have on the right side, you see this MinION flow cell. There the DNA is loaded on the cell. And you have a membrane which consists of the famous nanopores, which is a pore with a motor protein, and the motor protein guides DNA, single-stranded DNA, through this pore, where we have an iron current, and this iron current changes when the nucleotides went by, and every one of these 4 different nucleotides gives different changes in the iron current, and from this we can calculate the nucleotide sequence of the DNA we added to the flow cell. There are 3 different types of flow cells. The small one and cheapest one, the fungal, where you have around 226 pores and a theoretical maximum output of 2.8 gigabases. Then you have the medium one, the MinION flow cell, with around 500 pores and 50 gigabases of output. And then the high class, I would say, the high class one, the PromethION flow cell, with around 2,700 pores and 290 gigabases of theoretical maximum output. And you can use these flow cells on different devices. For the MinION and fungal, you have the MinION and GridION devices. For the PromethION devices. And we are using the PromethION 24, where you can run 24 of these flow cells at the same time. And we started several years ago now a pilot study with this technology, where we wanted to analyze the gut microbiome composition of hematopoietic of hematological or patients with hematological diseases, mainly leukemia, different types of leukemia. And these patients receive an allogeneic hematopoietic stem cell transplantation as a treatment, which is done from a healthy donor. Blood stem cells are collected, and then the unhealthy stem cells of the patients are treated with high dose of very aggressive chemotherapy, and they are destroyed, and the healthy donor cells are then transfused to the patients to build up a new stem cell and new immune system for this patient. And during this treatment, you have 3 different stages, which you can see on the right. Maybe I have also the pointer here. Yes, I have it. You have here the stem cell transplantation, HSCT. You have the phase before transplantation. You have the leukopenia phase, when the old stem cells and the old immune system of the patient is destroyed, and the new one is built up from the donor cells. And then you have the phase of the reconstitution, where the donor cells build up the new immune system for the patient. And during these time points, we collected stool samples from these patients and extracted the DNA and analyzed them with nanopore, some of them also with Illumina, but I will come to this later. We developed complex pipeline for the analysis of our nanopore raw data, where we did base calling, so the translation from the iron current changes to the nucleotide sequence, and then an alignment with the KMER-based tool Kraken-2, where we then calculated the microbial composition on species level for the stool samples. We also used the mapping tool Minimap2 to compare or to map our nanopore reads against the CART database to screen for antibiotic-resistant genes in the found species. And also we did for some samples Illumina sequencing, combined it with our nanopore results, and used this to look deeper into the microbial composition, not only on species level, but one step further on strain level. But I will come to this also at a later time point. So we analyzed our patients, and we also got 11 healthy control programs where we got stool samples from as a control cohort. And first, we just extracted the DNA and measured it, and we saw that the amount of DNA we were able to extract from stool samples from the patients was significantly lower than from our control patients, especially in the leukopenia phase. The total read number was quite stable because we were only able to put not more than 400 nanogram of DNA on the flow cell. Also, the medium lengths were comparable. The standard deviation for this was a little bit lower for the control samples. And then we analyzed the reads, and we saw that a fraction of around 60 to 80% of the reads were microbial reads, so bacterial, fungal, and so on. But we had also an amount of reads belonging to human DNA, so the host DNA, which increased during leukopenia and was lowest for the control group, which was convenient because during leukopenia they got aggressive high-dose chemotherapy, they got the antibiotics. This disrupt the gut membrane, and for this would be the explanation why there is an increased amount of human DNA in the stool. We found a constant amount of plant DNA, which was nearly the same fraction for all phases and also for our controls. This was mainly from food intake, so we found tomato and so on. So we excluded this from further analysis because it was not really part of the microbiome. But we also had a surprisingly large amount of DNA or DNA reads we could not classify with our methods, which was around 30% to 10 to 30%. This is the dark matter of the microbiome. Maybe these are some species which were not in our database. Maybe the quality of these reads was not good enough to analyze them with our database. In the future, we want to look at this because I think it's very interesting. Maybe we find something new here, but yeah, we have to develop methods to analyze these unknown dark matter of the microbiome. We then analyzed only the reads for microbial or microbiota, and these were the results. So we detected around 30 to 50 different species normalized in the control samples, and the number of species decreased significantly during leukopenia. On the same time, the amount of antibiotic resistant carrying reads increased. So this was we saw this effect of increasing of the antibiotic resistance and a decrease of the detected species. The largest fraction of this microbial reads belong to bacteria, around 90 to 99%. But we were also able to detect some fungal reads here in yellow, which interestingly increased in the leukopenia phase, while we have a slightly decrease of bacterial reads. We also detected viruses, only DNA viruses, of course, and also some archaea. Then we analyzed the microbial pattern our patients had before transplantation. So here you can see a principal coordinate analysis where we plotted all the stool samples, so from every time point. But in colored, there are only the stool samples taken before transplantation. So in the first 10 days before day 0. We see also we were able to detect a 3 cluster-like structure where we had this purple cluster here, where the patient samples look very similar to the control samples. So maybe I have to explain, in this principal coordinate analysis, the nearer the dots are, the more similar the microbiome composition is between these samples. So we have here a pattern of patient and control samples, which we defined as healthy because the control is in the same cluster. And this was mainly dominated by Bacteroides uniformis, but also Phocoeocola durae and vulgatus, which were in the past also Bacteroides. So we can say this cluster is a Bacteroides-dominated microbiome. Then we had this cluster 2, where we had not really domination of one specific species, and we had this cluster 3, which we defined as more unhealthy, where the microbial composition was dominated by Enterococcus faecium. At the same time, you can see here as this antibiotic resistant gene carrying reads, and we see an increase for cluster 3. So the unhealthy cluster has also more antibiotic resistant patterns than cluster 1. And we looked at the pre-treatment of these patients, and we could see that in these patients had a longer history of antibiotic intake, of longer treatment before. So this is highly convenient that these patients start more unhealthy in their microbiome compositions than the other ones. The question was now, does this has any effect on the transplantation and the treatment after the transplantation? And for this, we analyzed also the stool samples from the other time points. Here you can see again the principal coordinate analysis for the samples taken before transplantation. And here you have samples colored. So the colored samples are samples taken during leukopenia. And we see that we nearly lose all of the samples which have a composition of cluster 1, and it all shifted to the right side, to the more unhealthy composition. The color of the dots shows from which cluster they originate. So these were samples which started here, but now have a composition comparable to cluster 2 to cluster 3. And during reconstitution, we see the other way, so the effect the other way around, that they return to the more healthy gut composition, but not all of them. Some of them stayed then in cluster 2 or even in cluster 3. So yeah, there was not real trend. If someone starts healthy, he ends healthy, and so on. Unfortunately, the number of patients was not that high, so we could not see any significant correlation between the treatment and their if the patient got into reconstitution or not, and the microbial shifts. Yes, but just for comparison, we started here with a clear 3 cluster-like structure, and we ended here mainly on the left side, but yeah, not all of the patients got back to their old microbiome composition. The elegance of this method is that we can not only look at bacterial species, which you can see here at the top, we are also able to look at fungi, for example, and all the other species. So we are not just looking at 16S regions. And for the microbiome, we saw that even the patients, but even patients in especially patients in the cluster 3, showed an increase of Saccharomyces cerevisiae, which is Baker yeast. So it's not known as a very severe pathogen. It can lead to bloodstream infections, especially in immunocompromised patients, but it's normally it's not known as a pathogen. And yeah, it was interesting that this fungi was increased in these more unhealthy patients, so during leukopenia and in cluster 3. We also found some archaea, not very much, some Methanobrevibacter in cluster 1. To be honest, there is not much literature. We do not know if archaea bacteria, also archaea, are human pathogen or not. There is no known, as I know, known human pathogen archaea. So I think this would be interesting to look further into in the future to see if we can extract something from this. We also looked at the DNA virome. It is also possible to analyze RNA using nanopore sequencing, but not for our case. So here we just looked at the DNA viruses. And we see that for the healthy controls and also for cluster 1, an increase of the crust-like virus, and on the other hand, an increase of schooner virus for the more unhealthy patients. Also interesting finding, which was not described in the literature before. The question then arose if it's possible to that some of the bacterial species the patients had in their microbiome before transplantation can survive this process. So, for example, a patient has an Escherichia coli before transplantation, then we can detect this E. coli during the leukopenia phase, and then again it has an Escherichia coli in the reconstitution. Is this the same? Not only the same species, but the same strain as before. So does some of the bacteria could somehow survive in darm crypts, in gut crypts and then rebuild the microbiome, or are they all eradicated? And these new E. coli are from, or again, acquired from food intake. And then it should be another strain than before. When we look at the complexity of the bacterial taxonomy, we see we start with phylum, and we ended at the species level and analyzed the species level, but that doesn't stop. We have different pathotypes for Escherichia coli, and then for every pathotype, different strains, and we wanted to have this high taxonomic classification resolution of the strain level, and for this we used the combination of Illumina and nanopore sequencing. And we analyzed for some bacteria species, for some patients, if we have a strain shift or a stable strain. You can see here an example for Bacteroides vulgatus for these patients, 12 to 39. And they had so we could detect Bacteroides vulgatus before transplantation, then they were transplanted. And we could detect them after transplantation. And in yellow, you can see that there we detected a different strain before transplantation than after, and in gray we detected the same strain before and after. So we see we detect both. Some of them, for some patients we had a strain shift, for some patients we had a stable strain, and this, we could not really correlate this with the clinical outcome of the patients. So we tried to analyze these patients if they got a retransplantation, an acute or chronic graft versus host reaction, a relapse of the hematological disease, or the death of the patient. But it was not really clear that there was a pattern that if they keep the strain, they were more healthier than if there was a shift. It looks like this for the Bacteroides vulgatus, but when we look at the Bacteroides uniformis, we could not see this in a clear way. Also for Parabacteroides, we tried this. But for all the species we analyzed, we saw for some patients, we see a shift and some are stable. So we at this point only looked at the microbiome of the patients. It's also possible to look at the proteome, transcriptome, metabolome from the stool samples. Maybe we do this in the future, but with this microbiome data, we were able to publish these results in scientific reports. So we could develop a method for the characterization of microbiomes simultaneously. So we could look at bacteriome, archaeome, microbiome, and DNA virome. And this exploratory study of the cohort of the allogeneic stem cell transplanted patients showed that our DNA extraction method and our nanopore analysis pipeline worked. We identified this 3-cluster structure, which was new for us. And maybe with further studies and larger patient cohorts, we want to dive deeper into this topic. The good thing is, from this project, we developed the pipeline you can see here, where we can use our extracted DNA for sequencing. We have a step where we can deplete the host DNA and then use the residual reads for mapping against the database. We also have a step where we do a meta or pangenomic assembly of reads from the sample and build up specific genomes, compare them with the database, and kick or drop duplicates, just to be sure that we do not miss something. So this is our try to to get a more convenient look into the dark matter of the microbiome. And with this, we can analyze similarities, correlations, and the composition of different sample materials. Yeah, but as I told you, this is only a small part of information we got from our patients. We have a lot more. We have diagnostic imaging, we have culturing and resistant testing, we have the medication data, we can look at the methylation patterns of the human DNA, and we have vital parameters and clinical laboratory parameters. And these two we used for a different study, so the same patient cohort, but different data. This was mainly done by our informaticians, and they developed an algorithm for a dynamic prediction of the mortality risk of the patients using these data. So you can see here for an example patient, we used the data from day 0 to day 30 after transplantation. And for every of these days, the algorithm gives us a score if the patient is on low, moderate, or high risk. And this patient died after day 51. So here the algorithm worked out and predicted that there was a high risk after day 18. When you look at the bottom part of this figure, most of the patients which were alive after 100 days had a low risk during the first 30 days after transplantation, whereas the patients which died between day 0 and day 100, most of them had a high risk. So the algorithm works, which is really nice and gives us this traffic-like similar scheme to identify patients with high risk. We also tried to figure out on what kind of features this algorithm looks to determine if the patient is in low or high risk. And interestingly, the highest feature importance we found around day 15 to day 25, and there were mainly two values that were very interesting. The first one, the feature for inflammation in the bloodstream, which is convenient because it's a factor also the clinicians looked at. And the HST is a factor for urea, which give us a good hint how well the liver and also the kidney perform, which is also crucial for the for the patient and the clinical status of the patient. We were able to develop this dynamic risk stratification system. This was a random forest approach, which worked very nicely. It's also interesting that we could show that the most important features are features relying on inflammation-related blood work parameters, only these two. So yeah, that was interesting, and maybe we can also use this algorithm now for other patient cohort to see if it works not just for our UKD cohort, but also for cohorts from other clinics. Now just a short note to an side project which we started during the research for the hematological patients. And for this, I have to explain a little bit about the different sequencing technologies, but I will keep this short. So we started in 1977 with Sanger sequencing in the first generation of sequencing. Then Illumina came with second generation short read sequencing. And since 2014, we have Oxford Nanopore Technologie and long read sequencing, where the accuracy gets better and better over the last years. The problem is it's still expensive. A flow cell costs around 900 to 1,000 euro. And to reduce costs, Nanopore gives us the opportunity of multiplexing. So what you are doing is you have your high molecular weight DNA you want to analyze, and you ligate a barcode, a specific barcode to your DNA. In this case, there are 24, but there are also kits with 96 different specific sequences. And after this, you ligate the motor protein to the barcode. We need this to guide the DNA through the pores, I told you in the beginning. And then you can pool all of your samples. So in this case, up to 24 different samples on one flow cell, and you can distinguish them using the specific barcodes. And we did this, of course, we wanted to reduce cost as much as possible. And we also used different projects on one flow cell. But then we realized something strange. In this case, we had 4 different bacterial isolates, 2 E. coli and 2 mycoplasma. And we also had 2 samples, metagenomic samples, which were wound swabs, which consisted of around 99% human DNA. And we subtracted these human reads. And after this, the relative abundance looked like this. So around 50% of our samples consisted of isolate 1 and the other 50% of isolate 2. And this was strange, and we repeated this experiment and could show that was not a wet lab laboratory contamination. And then we thought about this. Maybe it's because of the barcodes we used and the technique of barcode ligation. And our theory was, so this is the normal protocol. You start with single separate samples, do the DNA repair, do the barcode ligation, then pool all the samples, and only the pool then is used for the adapter ligation, an enzymatic step where the adapter is ligated to the barcode. And normally you have this DNA-A with its correct barcode 1 at this step. But you can also have DNA-A without barcode 1 and also free barcode 2 from another sample. And because this is an enzymatic step, during the ligation of the sequencing adapter, it could happen that DNA-A with barcode 1 get the adapter, but also barcode 2 is ligated to the DNA with the adapter, and then you have the wrong barcode ligated to the DNA. To test this theory, we developed a modified in-house protocol where we kept the samples separate until the adapter ligation, and only after this we pooled them and loaded them on the flow cell. And we tested it with 4 different isolates and different concentrations. I can show you here. We used Escherichia coli in the highest concentration, Proteus mirabilis in a dilution 1 to 10, the Acinetobacter baumannii in a dilution 1 to 100, and Staphylococcus epidermidis 1 to 1000. And yeah, we analyzed them on one flow cell and with our own protocol on another flow cell. And the results were, as our theory expected. We saw an increased amount of incorrect reads in the Staphylococcus epidermidis sample, so the sample with the lowest amount of DNA. Here we found reads with the right barcode for this sample, but belonging to Escherichia coli, Proteus mirabilis, and Acinetobacter baumannii. And we didn't see this effect when we kept them separate until after the adapter ligation. During this time, Nanopore updated their protocol, so they noticed the same effect, and they changed it from an ethanol washing step during the barcode and adapter ligation to an SF, so short fragment buffer washing step during this. So we tested also this protocol with the same isolates and the same dilutions. And here are the results. So for the old Nanopore protocol, we have up to 2.4% of incorrect reads in our low concentrated sample. And this effect was dramatically decreased by changing the washing buffer, so from ethanol to the SFB. But still, there are some small fractions of incorrect reads left. The protocol we used, where we kept them separate, eradicated this effect completely. The problem is this is much more expensive because you need a lot of this enzymes because you have to use it for every single sample and not only for the pool. So this is much more expensive, but yeah, you have to decide if it's worth it or not to do it. I think it's depend on the project and the sample. But the new protocol from Nanopore is also very good version compared to the old protocol. So we could show barcode crosstalk is a problem for NGS sequencing. There is not much published about this. There's more about this problem for Illumina sequencing. And especially if we have these low concentrations of DNA on our flow cell. But the improvement of the protocols brings significantly better results. This work would not be possible with all these persons you see here. So I didn't do it all for myself. We have our working group in the medical microbiology, especially our head of institute, Klaus Pfeffer, but also Alexander Dilthey, Birgit Henrich, Colin McKenzie, Tobias Wienemann, and Anna Rommerskirchen, which also worked in these projects. On the other side, so we are more the clinical and wet lab part of the project. We have the dry lab or the informatic part where Philipp Spohr, Gunnar Klau, Max Ried, and Rebecca Fröhlich worked with us together as a cooperation in this project. And we got our samples from the Department of Hematology, especially from here we have to name Rainer Haas and Paul Jäger. And of course, without foundation, this would also not possible. So I want to thank the Jürgen Monchot Foundation and the Monchot AI Research Group for funding this research over the last years. Now I want to thank you for your attention. It's very important to don't mistake a min-ein for a minion. And if you have any questions, please don't hesitate to ask. Thank you.